We're still talking about aerobic cell respiration. I've got over here a nice computer graphic of a mitochondrion, a three-dimensional one, and I'm just going to explain you now the different parts of the mitochondrion and what happens where. You have to already understand the whole process of aerobic cell respiration in order to understand this a little bit better, but maybe this helps you visualize the whole thing. So that's a mitochondrion. So what do we have outside here? Outside here we have our cytoplasm, right? Because the cell, that's the cell, right? Yeah, and we have outside here the cytoplasm. I'm just going to erase this again. It looks kind of stupid. Okay, so in the cytoplasm, what we have in the cytoplasm is we have glycolysis. Glycolysis in the cytoplasm, and so in the process of glycolysis, we're taking glucose. Okay, I'm not going to talk about all these steps, and we make pyruvate, 2-pyruvate. Pyruvate, and these 2 pyruvates now go into the mitochondria, and in here we have that's the matrix down here. I mean, you see the label over here, right? It's the matrix, and in the matrix we have the link RXN reaction. Okay, and in the link reaction we make acetyl coenzyme A. Okay, so acetyl CoA, that is the link reaction. And I'm now drawing boxes here. I shouldn't have drawn a box here. Okay, because it's a substance and I'm not. Okay, so that we make acetyl CoA, and the acetyl CoA will then go into the Krebs cycle, which also occurs in the matrix. So we call it Krebs cycle, and this Krebs cycle also occurs in the matrix. And then the result of the Krebs cycle is a lot of CO2, of course, which is then given off and exhaled. But the Krebs cycle, we also produce a lot of NADH and FADH2, NADH. And this NADH will now go to the inner mitochondrial membrane. That's it. That's the inner mitochondrial membrane. Okay? Yeah. And what you have over here is you have all of these electron carrier proteins in here. And this NADH will now go into the inner mitochondrial membrane, and what will happen is we're pumping hydrogens across the membrane into the intermembrane space, which is in between here, between the membranes. That's the intermembrane space. Okay, but that's a membrane, and that's the other membrane over here, the intermembrane space. Yeah, and these hydrogens then go back over here to make ATP in oxidative phosphorylation or electron transport. Right, so that is kind of the rough overview over the whole process, kind of linked together with a drawing of the mitochondrion. So please remember, memorize what happens where. So glycolysis occurs in the cytoplasm, the link reaction in the matrix of the mitochondrion, the Krebs citric acid cycle in the matrix of the mitochondrion, and then the electron transport or oxidative phosphorylation in the inner membrane of the mitochondrion. Okay. So that is it, and I'm just going to erase this a little bit again, simply because I want to explain you some other parts here. Not everything which is in this drawing here is relevant, okay, but there are a few things that are relevant. Yeah. So we already have talked about this one here is the outer membrane of the mitochondrion. This one over here is the inner membrane of the mitochondrion. This here is the intermembrane space. I'm just going to write it over here, okay. This down here in here that's the matrix, okay, of the mitochondrion. And these infoldings over here, they're called the cristae. You got it over here, okay. The cristae are these infoldings, and they increase the surface area. Okay. You also see that there are ribosomes here. They're so-called 70S ribosomes, like bacteria. They have bacterial ribosomes, and you see over here the ATP. Synthase, the one that you already know, right? Okay, and you also see that mitochondria have their own DNA. It's mitochondrial DNA, pretty small, but still they have their own DNA. Yeah? And and that is it. So you please know the structure of the mitochondria, how the parts are called, and also what happens where. So yes, next one. What I'm going to do now is I'm going to give you yet more simplified aerobic cell respiration overview. So that's actually you can fill this in yourself. Really, I'm just going to label the boxes. That's called glycolysis. Okay, this one over here, that's the link reaction. What do you think? This is a Krebs citric acid cycle. Krebs citric acid cycle And this one is the electron transport or oxidative phosphorylation. Phosphorylation means addition of phosphate. Yeah. Phosphorylation. Okay, I'm getting out of the box here. So, and what I'm doing right now is I'm just going to give you a big overview. We start with glucose. We are adding glucose to glycolysis and we make 2 pyruvates. Okay. In the process, we're making also 2 ATPs net, and we're making also 2 NADHs. Okay, and we have 2 pyruvates. The pyruvates go into the link reaction, and in the link reaction, we're producing for each pyruvate a CO2. We're adding 2 pyruvates. So how many CO2s are we making? 2 CO2s. We're making also 2 NADHs. Okay, oxidative decarboxylation, 1 CO2, 1 NADH for each pyruvate. The end product of the link reaction is acetyl-CoA. How many acetyl-CoAs do we make? 2 acetyl-CoAs. 1 for each from each pyruvate. CoA. So 2 times a C2 substance. Okay, 2 times a C2 substance. And the acetyl-CoA will now go into the Krebs cycle. And the Krebs cycle will now run 2 times, once for each acetyl-CoA. So actually what I have to do is I have to draw, indicate here it runs 2 times. That's important, okay? It runs 2 times. And what we're making is we're making a lot of many NADH. Well, actually I have to be now very careful. I make, how many NADHs do I make in total? I make 1 from the first oxidative decarboxylation. Okay, 1 for the second the oxidative decarboxylation. 1 when I'm oxidizing the C4 substance. So that means I make per acetyl-CoA I'm making 3 NADHs multiplied by 2 because I'm adding 2 acetyl-CoAs. 3 times 2 it must be actually 6 NADHs. Okay. Then I also make 1 per acetyl coenzyme A. I'm making 1 FADH2, so I must make 2 FADH2, and for each acetyl CoA I'm making 1 ATP, so I have a total of 2 ATPs which are directly useful. Okay, so that is what I'm making. And what happens to the carbon dioxide? Well, yeah, for each acetyl CoA I'm making a CO2 times 2, 4 CO2s. Okay, because it's running twice. 2 carbon dioxides per cycle times 2 makes 4. Okay, so let's see if I'm actually so far still correct. So 4 plus 2 makes 6 because glucose is a C6 substance. Okay, so far I'm fine. The question is now what happens to all of this stuff here? The FADH2 and the NADH, now they go into the oxidative phosphorylation. Okay. I need oxygen, 6 O2, 6 molecules of oxygen, okay, to make 6 molecules of H2O. And what is the whole point of this? To convert the energy into a lot of ATP, and around 38 ATPs. So I got to add 36 ADPs plus 36 inorganic phosphates, and to make, no, not 36, 38. 38, my mistake, because 38, so, and then we have 38 ATPs, and because we're making per molecule of glucose around 38 ATPs, but we've invested here, which I didn't show, okay, we've also invested some ATPs here at the beginning, so essentially we are down to 36, and now I'm just seeing that actually maybe we are making only 36, because over here we already made 2 ATPs. Okay, so wow, you know what? How many do we now make? 36 or 38? I'm confused. I don't know what the correct answer is. You know what? Neither do the scientists. Okay, so how many is it now? What I read is that the scientists indeed are still disagreeing a little bit on the exact number of ATPs produced per molecule of glucose. It is around 38. Okay, the reason is why you cannot completely convert this one-to-one is because in oxidative phosphorylation, it depends on the number of hydrogens that are streaming across the membrane to make a molecular ATP. What I read somewhere as a good how shall I say, average is that we're making around a total of 38 ATPs per molecule of glucose, but we're taking away 2 at the beginning for glycolysis, so we're making around 36 net. Okay, 36 molecules net ATP, yeah, per molecule of glucose. So that is the overview. Okay, wow. So we've got 1, 2, 3, 4 important stages. Okay, and for the sake of completion, I'm just not gonna also tell you that over here the pyruvate can also go the so-called anaerobic pathway. Anaerobic. Please read the standard level part where I'm fully explaining the anaerobic pathway. Okay, or not watch the standard level videos. Okay, because over here in the anaerobic pathway we don't need oxygen, but over here we do need oxygen. This is super important, this thing here, because the oxygen is our final electron acceptor, and without that we cannot do oxidative phosphorylation. Without oxygen, without oxidative phosphorylation, we cannot do the Krebs cycle. Yeah, we cannot do the link reaction. Yeah, the whole thing here cannot take place without oxygen, and then it's going over here into the anaerobic pathway, where we are at least making a little bit of ATP over glycolysis. So the anaerobic pathway would go like this, and at least we're making a little bit of ATP. So I think that's enough for right now. I wish you all the best.